Okay, so last time we ended this class, we ended with what a fatty acid was. And we were talking about lipids and how lipids are put together. Now, we're going to continue that model. And you recall this from class, we have this fatty acid model right here. And a fatty acid model has multiple carbons with hydrogens on the side. And on the end, we have that carboxylic acid, that C double bond O OH. Okay, and this is one of the functional groups that you had to know for the first test. Now this one I've drawn to the right is indicated right here on your diagram. Now what's going to happen is we have another molecule called a glycerol molecule shown right here where I've drawn it. And you see how we have an OH on that glycerol molecule. Well, I'm going to draw that carbon and that OH right here. And this is what we call an alcohol. And that's another one of those functional groups that you had to know. So what's going to happen is the oxygen and hydrogen from this carbon and the hydrogen from the glycerol are going to come off as a water. Now, as this comes off as a water, do you recall we talked about the idea of a condensation reaction or a dehydration synthesis? All right, this is your condensation reaction. As these two come off, it will form a covalent bond here. All right, and this covalent bond that is made will now link the two molecules together. Now, if you could imagine what's going to happen here, this fatty acid, okay, shown in the purple on your screen, this fatty acid will connect covalently to this glycerol molecule. All right, that would be a monoglyceride. Now, what would happen if we added another and then finally a third one? We would have a triglyceride. All right, again, triglycerides are when we add 3 fatty acids to a glycerol molecule. We do this via dehydration synthesis or a condensation reaction, and water is going to come off of this as we do this. Okay. Now here is a triglyceride, and this is an example of one of these we just talked about. Now this is a triglyceride. Here we have 3 fatty acids connected to a glycerol backbone. Very important for storing energy in the body. Okay, we're going to store a lot of our fats in this triglyceride form. And that, as the name implies, we just talked about this, we have tri, meaning that we have 3 of these fatty acids connected to our glycerol molecule. Now, I want you to notice a few things. Do you see after every one of these, we have an H2O, an H2O, and an H2O? What this is saying is that to connect this to the glycerol, we had to remove a water, a dehydration synthesis, or as we called it before, a condensation reaction. Another thing I want you to note is, do you see how we have this double bond here? It looks like a crook, right? We talked about this before, and that was a cis double bond, right? This is an unsaturated fat, right? A mono unsaturated fat, mono being 1, right? And anytime we have a double bond, we have an unsaturated fat. Okay, so that is a triglyceride. Okay. The next is a phospholipid. Now, we're going to come back to the phospholipids when we talk about the membrane. Very similar to the one we just talked about, in which case we have here, same idea. We have a glycerol backbone. All right, so we have that carbon, carbon, and another carbon. Now, before we had what? We had 3 fatty acids on this, right? That was our triglyceride. Well, we're going to have a similar structure here. We're going to have, I'm going to draw this line. This is a fatty acid hydrocarbon tail, and here's another of those hydrocarbon tails we just drew. But instead of having a third one, here we have a phosphate-containing region. All right, it has phosphate or phosphorus and has oxygen here and some other things. Point is, and we'll talk about this in another class when we talk about the membrane and the structure and how that integrates with phospholipids. But what I want you to know now is there's a dual nature here, okay? That these tails are hydrophobic. They are afraid of water, so they hate water, okay? Now, we'll talk about this in class later on, how these behave. But these are going to, if you will, hate water. They don't want to associate with water. The head here is polar, and it loves water. I'm going to put a smiley face water. Okay, this polar head loves water. And this dual nature of having a polar region here and a non-polar region here will be important in how our membranes organize. Okay, for now we're just going to identify it as a particular type of lipid so you know the structure. We'll talk about how these behave in water later on when we talk about how membranes organize. All right. Another type of lipid you need to know are the steroid or the sterile rings. Okay, these are the basis for many things we have in our bodies. For example, your testosterones, estrogens, cortisols. All right, you've heard of testosterone. That's a type of hormone, right? Same thing with estrogen. That's another type of hormone. Well, it's based on this 4-carbon ring structure. So do you recall what we said that lipids were rich in? What are lipids rich in? They're rich in carbon, and what else? They're rich in hydrogen, all right? And what are they poor in? They're poor in oxygen, all right? Now, what are these? Look at all of these structures. Each of these are really rich in what? Well, recall these are structural models, right? And remember, at each little bend right here, we have a carbon, right? So we're seeing that there's a lot of carbon here. We're seeing that there's a lot of hydrogen here, right? What are we seeing there's very little of? That is oxygen. We're not seeing a lot of oxygen here. We're seeing a lot of the carbon and hydrogen, but not so much of the oxygen. And this is, why we would link these in with the lipids. Now, another one you're probably familiar with is that of cholesterol. We'll come back to cholesterol later on when we talk about its role in the membrane. It does have a role in our membranes, but for right now, it is just to understand that it's based on the sterile ring structure. So let's look at some specific examples of this. So for cholesterol, really important in our cell membranes. Okay, it's going to change the fluidity, how our cell membranes move around. Again, we'll bring that more into the forefront when we talk about membranes later on. Another example are steroid hormones. Okay, the second point right here. There's protein hormones and there's steroid hormones. Many or several hormones are steroid based, and these are grouped in the lipids. Okay, you've heard of testosterone, estrogen, cortisol are grouped into that grouping. Also, bile acids. Now, you're going to come back to the second point. Let me pull back here a second. Next semester, the second part of this course, you're going to talk about how hormones work. And you're going to talk about how steroid hormones work and how protein-based hormones work. So you're going to see this in the future. All right? And then bile acids, another example of these compounds. So obviously, this is a fairly important structure. Now, for the purposes of the test, you really should recognize this type of ring structure. You do not need to memorize, for example, the difference between testosterone and cortisol. Okay, you don't need to commit this structure to memory, but you should be able to recognize this 4-ring structure as a sterile structure of steroid-based hydrocarbon. Okay, so what are the possible roles of protein here? The first role is a structural role. Think about your cells, collagen, your hair, keratin. You're made of protein in many ways. Okay. Another role of protein is regulatory. Many of your hormones, insulin, glucagon, are some examples. These are based on, you guessed it, protein. All right, contractile, actin, myosin. When you start seeing how a muscle works in the sliding filament model that you'll see next semester, again, protein-based. Immunological, think about antibodies. What goes after those pathogens in you? Those are protein-based, and you'll see that next semester. Transport, think about your hemoglobin. We have 4 separate protein chains. Again, protein-based. Many or almost all of your enzymes are protein-based. So very important to talk about proteins. Now, this is the core shape of an amino acid. You have to know the generic amino acid shape for the test. You have to know this. All right, think about what you know of an amino acid, right? Amino is this group. We have a nitrogen and some hydrogens. Now, depending on the conditions, it can take on an additional hydrogen. That's why you see 3 hydrogens here. But on one side, we have an amino group, and it can take on hydrogens. On the opposing sides, we have a carboxylic acid group, a COOH group. And this is why we call it an acid, right? We have an amino acid. This is the acidic group. In the center, we have an alpha carbon, and attached to this, we have an R group, all right? We'll also call this R group a side chain, and it is the specific R group or side chain that makes a specific amino acid. For example, why is tryptophan tryptophan? Is because it has a specific R group. Why is glycine glycine? Again, it has that specific R group. So this R group defines what an amino acid is. And this entire structure shown here is an amino acid. And I'm going to use just the letters AA to represent amino acid. And these are going to be the building blocks of the protein. These are the blocks, the monomers, right? And then the protein is like is essentially like a wall. Or maybe a better analogy would be these are like a link on a chain, and the entire protein is like a chain connected with many links. Okay? Now, again, you have to know the specific groups on one of these amino acids. You have an amino group, and we talked about you having to know this functional group. We have a carboxylic acid group, again, COOH, or here COO, and it's just based on the pH that this is kept at. And also we have that side chain, or that R group. Again, you have to know this general structure of an amino acid and these functional groups that we have discussed previously. Now, as I said, this R group defines what an amino acid is, how it behaves. How does it behave in solution? All right, so let's look to the right, and as you see, we have what? We have a blue zone on all of these, right? That in H2 we've been saying. And then we have a red zone over here, that COOH, that I have also been discussing. Now, these are those amino and carboxylic acid groups. What's different in all of these? We see that the difference is what? It is that R group. So here we have glycine. Glycine, that R group is a hydrogen. Okay? Tyrosine, that R group has a benzene and an OH group. All right? Lysine, we have several carbon hydrogens and a nitrogen group here. Again, the R group is different. Cysteine, again, we're going to have a sulfur group involved in it. That's going to be important for disulfide bridges, and we may get into those later on. But what I'm trying to impart in you now is it's this R group, or we call it a side chain. That is what varies in these structures. Okay, that is what gives an amino acid its chemical properties. And because because a protein are many amino acids connected to one another, these R groups, or these side chains, also gives a protein its characteristics. Why a protein takes on the shapes that it does all goes back to what side chains we have. Now, how are we going to connect these? Remember, I've said that amino acids are like links, and then all those links together are a chain. Okay, how are we going to connect those links? Well, how we have connected carbohydrates, how we have connected fatty acids to a glycerol, same thing here. We have that condensation or dehydration synthesis, same thing, reaction occurring. And when it does, we form a peptide bond. Okay, so how did this happen? Here we're going to remove the OH from that carboxylic acid, and we're going to remove a hydrogen from the amino group, the amine group. Those are going to come off as a water. And when those come off as a water, I'm going to draw that water over here. All right. When those come off as a water, we form a peptide bond. And when we form this peptide bond, that is what connects these two amino acids. Now imagine the same thing playing out hundreds of times. All right. What's going to happen? You're going to connect another amino acid here. You're going to connect another amino acid here, and you're going to connect another amino acid here, and then another amino acid here. So you're going to do multiple reactions like this, and this will form a protein. Okay, but again, it is all just several dehydration syntheses or condensation reactions stacked on top of one another. And they together form, and this is something you have to know, is a peptide bond. That peptide bond connects two separate amino acids to one another. All right, and again, dehydration synthesis, same thing as we have stated before. You have to know this concept of a peptide bond. It is the bond that connects two amino acids. Multiple peptide bonds are needed to make a protein because we are connecting multiple amino acids. Now, something I also want to draw your attention to is, and we've covered this previously, is that idea of an R group. Do you see how we have glycine? Glycine, what's the same with glycine and alanine? Well, their amino groups are about the same, right? Their carboxylic acid groups, same idea. What varies between these two? The R group. Here, alanine is a CH3. All right, glycine has a H for an R group. So they have different R groups. This makes them different amino acids. All right, so I think we've pretty much covered this very well. But again, emphasis on this, the peptide bond. All right, that is what connects two amino acids. Now, how are we going to break this peptide bond? How did we break other bonds? Recall, we have talked about this with carbohydrates. How are we going to break that bond? We're going to do hydrolysis or hydrolysis. You can call it either one you want to. I like to emphasize the hydro. When you think of hydroelectricity, for instance, you think of, well, obviously you're going to think of water, aren't you? All right. When you think of lysis, as we said in class, what do you think of when you think of lysis? Well, in biology, lysis is basically shorthand for break. All right, so we're going to break something apart with lysis. Well, same thing here. We want to break this peptide bond, so we're going to go this direction. All right, here we have this peptide bond, and we want to take this peptide bond and we want to break it. Okay, well, as we talked about before, when we can make that peptide bond, a water comes off, right? So guess what we have to do if we want to break the bond? Well, we have to break lysis, hydro, water, so we're going to have to break a water. We're going to have to split a water, and one part of the water, the hydrogen, okay, will go with the amine over here, and the OH, or the remaining part of that water, will go with the carboxylic acid, and that will split the bond. All right, so now we're talking about how we break these peptide bonds. And think of this as on a biological sense. You have to, sometimes you're going to break proteins down for energy. Okay, it's not a prime energy source, but it can be involved in metabolism and energy conversion. Well, you're going to have to break these bonds in order to release energy from them. All right, so this does become important in our bodies. So we're going to cover this in a separate video, but for now we're going to also talk about it here because it's a very important concept. Now, I've made a separate video of this, and I'm going to also cover it in these slides, and it's important to understand for a few reasons. First is when we start talking about how we go from DNA to RNA to protein, this becomes important. Another reason this is important is when you start thinking about how the genetic basis for disease, when you have mutations in a gene, for instance, this can become important because sometimes that mutation will introduce another amino acid, change amino acids around, and that changes the entire function of a protein. Now, this is not the only way a mutation happens. There's other ways, but sometimes we're going to change an amino acid, and when we change that amino acid, we change how a protein functions. And this goes in explaining how that works. So this is a very important concept to understand. So we're going to start with primary sequence, or the primary structure. The primary structure is the linear order of amino acids. So as words are in a sentence, that is the primary structure of amino acids. So for example, we have some sentences here, right? Amino acids are connected. Think of each of those words as a separate amino acid. And you see how there is a linear order to them? And again, what are they connected with? Peptide bonds. Remember that from the last part? Okay. We connect these via peptide bonds. Now, how these are ordered is the primary structure. Now here we just have a circle for each R group. Okay, we don't have a different chemical structure, because for space considerations. But imagine each of these R groups as being different. Okay, and the specific order of the amino acids as they are linked together with that peptide bond, wink wink, hint hint, as they're connected with that peptide bond, that is the primary structure to an amino acid. And this primary structure, as we will discuss in the next test, all goes back to the DNA sequence. The DNA sequence leads to the primary structure of an amino, or of a protein. Very important second point here. I want you to highlight this, star this, circle this. The primary structure, the sequence of amino acids, is important to determine all other levels of protein structure. Very important concept. So, Shakespeare, I believe, said the play is the thing. Okay? And in that context, sure. Here we're going to say in how a protein functions, the shape is the thing. How a protein is shaped is very important in how a protein functions. It is a critical point. How that protein is shaped is how it functions. For example, if we have a lot of secondary structure in a protein, for example, a lot of alpha helices, that protein very well may be a transmembrane protein. That's just an example. Don't have to memorize that for the test, just an example. So how a protein functions is very interrelated to the shape of that protein. And the shape of the protein is important because the primary structure, the primary sequence of the amino acids, the order of the amino acids, is important in determining that structure, that three-dimensional shape of the protein. Okay, so primary structure, leads to shape. Shape leads to function. Okay? If you were to change the primary sequence, you may change the shape. If you change the shape, you may change the function. Now, I'm using the term may here because does it always? No. Or if it does, it's such a minor amount that it's inconsequential. Depends on the change. Sometimes that change can be significant. Sickle cell anemia, good example. One change in the primary structure, one single amino acid is off. And because of that, the entire function of that hemoglobin molecule is altered. Okay, so it can become very important. And it's more of a case-by-case basis on that, and we will get to that in later chapters. Now, the primary structure is determined by the DNA sequence. And more on that later. Again, we get to that in later units of this course. But for right now, I want you to know that the sequence, the linear order of amino acids, which amino acids link up, is dictated by the DNA in our cells. So that will determine the specific amino acid sequence. All of this is ultimately determined by DNA. We will get to this point when we talk about the central dogma of molecular biology. You will also cover this in your laboratories for this course. But for right now, I want you to know that the DNA sequence, okay, codes for the primary structure of a protein. And this is the first level of organization. The next level of organization is secondary structure. Now, before the primary structure, again, was the sequence of amino acids. And I'm using AA to represent different amino acids. And we made the point that the specific sequence, the actual sequence of these amino acids, was the primary sequence, and you recall that these were connected with what type of bonds? Well, play the Jeopardy music. That would be peptide bonds. All right, so each of these horizontal lines is a peptide bond connecting them, made with, as you recall, a condensation or dehydration synthesis. Now, fantastic. With this being said, can these amino acids also interact with non-adjacent amino acids? And the answer to that is yes, they can. For example, this amino acid right here may interact with this amino acid over here. It might form a hydrogen bond. And this amino acid right here may form a hydrogen bond with this amino acid over here. And this amino acid right here can form a hydrogen bond with this amino acid over here. So this is to say that non-adjacent amino acids can interact with one another. And this interaction, for example, hydrogen bonds, can form these secondary structures. Now, we talked about this in the other video. This is a helix. Okay, so think of this as almost like a curly fry looking shape, or a spring shape. It's a windy circular shape here. Okay, think of it like a curly fry. And along this, we have what? We have an R group, so we have an amino acid here, we have an amino acid here, we have an amino acid here. So all these R groups are indicative of a amino acid, or an amino acid. All right, now what's happening here? Do you see how the one amino acid here is bonding to the other one here via a hydrogen bond? Do you see how this amino acid up here, I believe this would be let's call this one 1, 2, 3, 4. See how this first amino acid right here is hydrogen bonding to this fourth amino acid right here. Do you see that? Do you see how these amino acids are interconnected? Okay, when they start forming that, they are forming hydrogen bonds, and this starts to the totality of all these hydrogen bonds starts to form these helical structures that we call alpha helices. Okay, and these are formed by hydrogen bonds between the different amino acids. Another form that can form is a beta pleated sheet. Same idea, but a different kind of configuration. Instead of being a curly fry type of shape, here we have a more plain shape, a planar, flat shape. Again, same idea. Do you see how here is the oxygen in our acid group? And see how I have a hydrogen in our amino group? They're forming a hydrogen bond, another hydrogen bond, another hydrogen bond, another hydrogen bond. These are two separate chains, or two chains of amino acids. So we can form a flat structure called the beta pleated sheet. So that is secondary structure. Now shown below, here's a good diagram of it. You see how we have this circular pattern right here? See how we have that curly fry looking thing going on? All right, that is an alpha helix. Do you see how we have this flat area here? Okay, beta pleated sheet. Again, right here we have that curled area here, an alpha helix. So as you see, the protein will have different regions of this secondary structure. And the secondary structure is formed by the interactions of different amino acids. For example, hydrogen bonds. And this will help form these structures on the greater protein. Here's a better diagram of those secondary structures. Now again, here on the left is our alpha helix. And we're going to kind of go around here. Okay, see how we have that thing going on, that curly fry spring kind of shape going on. We see that? All right. So think of this as kind of coming in and out at you. And you see here is our first amino acid right here. Here's our second amino acid right here. Here's our third amino acid right here. And here's our fourth amino acid right here. Do you notice how the first amino acid is forming a bond? This is a hydrogen bond with the first amino acid. Again, the interactions between these amino acids. And we can continue this discussion, 4, 5, 6. And you see how the second and the sixth one in this diagram is forming a hydrogen bond. Point is that non-adjacent amino acids in the primary sequence are interacting with each other, forming these hydrogen bonds and thereby forming these structures that we are calling secondary structure. The one I just described is the alpha helix. All right, trying to show you some three-dimensionality here. And the one right here is that beta pleated sheet. Same idea, we're hydrogen bonding between two amino acids, and that hydrogen bonding will make a discernible structure on the protein. That is secondary structure. Tertiary structure is the overall three-dimensional shape of the protein. So here we have, again, our secondary structure. Alpha helix here, some alpha helix here, some random coil here and there. We have some beta pleated sheet here. Okay, the entirety of all of the secondary structure, all of it together is the tertiary structure. Okay, so the entire thing, all the coils and runs and beta pleated sheets and everything combined in one big large molecule, we describe that as the tertiary structure of a protein. Very important idea here, that protein shape equals function. This is a organizing theme when we start talking about how enzymes and how proteins in general work. Remember I said, Shakespeare said the play is the thing? Well, in biology, it's the shape is the thing. What shape a protein is very important in defining its function. All right, very important concept. Why is this important when we start talking about primary sequence? Well, what helps define the shape in these interactions, you can ultimately go back and say it's the primary sequence, the order of amino acids, that helps to determine the ultimate shape of a protein and its function. And you might ask the question, why is this important? Well, what if we could change the primary sequence, right? So we have different amino acids. What if we changed this amino acid? That change may, I'm not saying it will, but may change the shape of that protein. That change in shape may change the function of that protein. All right. Now, what leads to this sequence of amino acids? What leads to the primary sequence of a protein? Well, that, as we said before, was DNA. So if we have a change in our DNA, if we have a mutation in our DNA, for instance, that may lead to a change in the primary sequence, the order of amino acids in a protein. That may change the secondary structures and consequently the tertiary structures in a protein, and that may change the function of that protein. Quintessential example of this, I've stated this before, is that of sickle cell anemia, because I like using that example because we've heard of that before. All right, we change one base. There's one base, one single nucleotide in our DNA or in DNA that can change. That one single nucleotide changes one amino acid. That one amino acid change changes the conformation or the shape of that hemoglobin protein. That change in hemoglobin protein changes the entire function of how that hemoglobin protein works. And it all goes back to one single change in the DNA. Now, we'll come back to this for future tests, but you can see how we're laying the groundwork for that. But very important to understand that protein shape is important in defining the function of a protein. The final shape of a protein, and many proteins are going to stop at tertiary structure, but sometimes a protein will have to combine with another to make a functional unit. And in that situation, we have quaternary structure. This is when two or more proteins combine, or in this case you call them polypeptides, to form a functional protein. Quintessential example of this is that of hemoglobin. This molecule you see right here, we have our red blood cells are chock full of millions of these, and these are responsible for moving oxygen from your lungs to your tissue. Now, do you see how we have four separate chains? We have two beta and two alphas. I'm just going to draw one right here. This is our first chain. Here is our second chain. Here is our third chain. And here is our fourth chain. So we have four separate individual polypeptide chains that are interacting with each other, that are linked, and this linking is defined as the quaternary structure. Now again, not all proteins will have this. Not all proteins have this type of situation. This is seen in some, but not every protein. Not all proteins. All right. In the example right here in the lower right-hand side, we see that we have tertiary structure here, and they're trying to show that this blue protein over here, this blue polypeptide chain, interacts with the gray polypeptide chain, and the entirety of that is quaternary structure. That completes our discussion about the third macromolecule, and that being protein. Now, we're going to come back to these nucleic acids and nucleotides later on, but this is the fourth and final one for the series. Okay, nucleic acids and nucleotides. Nucleotides, these are the monomers, all right? These nucleotides will combine, and these are nucleotides right here, so this is a nucleotide, and this is a nucleotide. These nucleotides combine to form our nucleic acids, and you know these as DNA, and you may have even heard of its, if it's a little pal, RNA. Okay, these are our nucleic acids. Now, we're going to come back to these a lot more when we start talking about how DNA functions. We'll come back to this concept. But right now, let's leave it with this, that nucleotides are the monomers. Remember we had that discussion about monomers and polymers? All right, and several nucleotides combine. Here's nucleotides. All these nucleotides are combined. And we form our nucleic acid. So several of these nucleotides combine to make our nucleic acids, such as DNA and RNA. Key point, nucleic acids are a polymer of nucleotides. Now we will definitely get more in-depth into this later on. Okay, you should also recognize that this is a nucleotide. So you don't have to memorize the base structure here, but just recognize that if you see something like this, this is a nucleotide. Okay. Now, what does a nucleotide contain? Has a couple parts here. We have a nitrogenous base. Okay, means it has nitrogen in it. So here is you've probably heard of A, T, G, and C. Okay, those A, T, G's, and C's are our bases, and they're chock full of nitrogen. Do you see how we have a nitrogen here and a nitrogen here? And lots of nitrogens. We also have a lot of hydrogens here as well. Okay, so we got nitrogens. I'm drawing blue dots around the nitrogens. We have, because of the nitrogens, remember we talked about hydrogen bonding and a very electronegative atom and hydrogen? Same idea here. Our DNA is held together by hydrogen bonds between nitrogenous bases. Okay, so these are hydrogen bonds right here and right here. So our DNA is held together by hydrogen bonds. Or the different strands are held together by hydrogen bonds, I should say. Okay. Another feature here is we have, remember we talked about carbohydrates? What was the ending that we associated with a carbohydrate? If you just said OSE, like glucose or maltose or sucrose, you are correct. Here we have a deoxyribose, OSE. What does that imply? That implies a sugar. So we have a carbohydrate as part of this. Our DNA base here is connected to this deoxyribose sugar. And then we also have on the end a phosphate. Okay, so have a general idea of the structure of a nucleotide. Now, you're not going to be required to memorize the structures of each individual base. You know, we're not going to get that level of detail, but you should be able, if you saw this on a test, you should say, bang, that's a nucleic acid. And you should be able to say, hey, this right here, that is a nucleotide. And you should also recognize that there are hydrogen bonds that connect these opposing nucleotides.